As most of you know, the rationale of this project and the main objective has been to design new nanomedicines for delivering therapeutic peptides through the oral route. And for that, we have defined some key properties for these carriers, which would be these, like they should provide an adequate stability in the gastrointestinal fluids. They should also favor the interaction of the system and a peptide with the mucosal barrier, and also to protect and to provide controlled delivery of the associated peptides. And our strategy to obtain this has been to develop a large number of nanocarriers based on well-known and safe biomaterials, mostly lipids, polysaccharides, polypeptides, and the rational combinations of these, eventually also combined with penetration enhancers or enzyme inhibitors. Then we had to design the adequate size and the surface composition for these carriers to provide these characteristics. And also we developed different structures, as you can see, lipidic carriers like nanocapsules, polymeric nanoparticles, and micelles or different self-assembled systems. And then in some cases, these were further incorporated in the simple technology, which is the final dosage form for some of these carriers. Okay, so this is just to show you where this work package is. This work package 1, where what we had to do is to have a wide range screening of the different carriers. You can see that at this stage we developed more than 60 different prototype compositions, and the objective of this work package has been the systematic screening of these to establish the selection criteria and decide which of these large number of carriers can go further in the project. So in this step, the major selection criteria has been the stability of the system, both during handling, storage, and also the colloidal and the biological stability in physiological fluids like different simulated intestinal media. And there, what the major challenge has been to take into account the effect of enzymes, surfactants, and lipids, and so on, the presence of these in these very complex biological media. And to allow comparative screening of these carriers, what we did is to design and approve between all the partners of this work package specific procedures, specific protocols, so that in the end we could have reproducible and comparable data. These, in this stage, we had 4 SOPs to characterize the systems, like the particle size, the surface charge, also the long-term stability has been one of the critical items, and we also developed some guidelines for the freeze drying of the nanocarrier, so that the handling and storage stability could be assessed both as a suspension or as a freeze-dried powder formulation. Well, from this stage, from this screening stage, what I would say is that it was very important to have a reference center for these studies, for all of these procedures, to assure that we really have the same conditions and we compare the systems and we can make a rational selection of them. And as I said, it was very important to assess the influence of the components of these very complex biological media. And our conclusion has been is that it's okay at this stage to assess particle size and surface characteristics, but then we have other factors that have to be evaluated later on in other work packages, like, for example, the loading of the peptide, or when we label the system with a fluorescent marker, these can also, in a way, alter the characteristics of the system. So what we decided is that even though the screening was really flexible and adaptable to all carriers, is that it needs to be complemented within the same work package with some other techniques like microscopy, or when it's a lipidic carrier, for example, lipolysis, or moving to the next work packages to assure that the protein associated is not degraded. Even though we can confirm particle size and surface charges unaltered, it's important also to assure that the protein that is associated to this carrier is still intact. Okay, so another important point has been the evaluation of muco interaction. We heard quite a lot about this in the first talk, but within this work package, what we did is first to develop some quick, not very specific, but quick and easily adaptable techniques, like, for example, the interaction of the carriers with the mucin solution, or to use a mucin film, like in a coated Petri dish, or other partners developed a similar system in a transfer plate, so that we could see the muco adhesion. For the first system, I would say that this is the only one, even though quite non-specific, but this is the only one that did not require the fluorescent labeling of the carrier, so this was a big advantage and allowed for a quick screening. All the other systems and techniques that I'm commenting require the fluorescent labeling of the carriers, and as I said, this can bring some difficulties to define just one technique that would be appropriate for all the nanosystems. Okay, so then we had for mucodiffusion, we worked both in mucin, different mucins, and also in mucus from animals. And then we had this microscopy evaluation with different systems. We have an SOP designed for this microcapillary technique when we see the diffusion of fluorescent nanocarriers in this mucus or a mucin filled in the microcapillar, and a similar system was developed based on a microfluidic device where we also have porcine mucus, and then we inject the carriers, and then we can see again the diffusion. You could see the diffusion of the particles within this mucus. In these cases, you see it's already more complex, also more specific system. What we found difficult is to establish a standardized way to inject. As we see velocity here, it's very important how we inject the system within these devices. Okay, and finally, the more sophisticated techniques that we used is the FRAP and the multiple particle tracking. I think this video probably won't work, so I won't try. And in this case, what we can determine is the diffusion coefficient after photo bleaching, for example, or in this case, in the multiple particle tracking analysis, what we do is to follow each individual trajectory of the particles, and we define the main square displacement of each one of them in a mucus or mucin solution. And regarding these techniques, well, it's just an overview to show you more or less. We developed 6 groups of different systems, ranging from the very fast and easy but not so specific screenings with their advantages and their limitations. And we also have the number of prototypes tested here so that you can make a comparison. Obviously, these last techniques are more complex and more expensive, and in this case, the reference center was very important to carry out these studies. And just to mention that these are all in vitro assays for the screening of the plant prototypes, and later on these have been also completed with the different cell culture, ex vivo and in vivo methods to confirm these first results. And the general remarks would be basically this, that we have a wide range of technologies, and it was really critical to standardize, like the mucus rheology, the origin of the mucus, really the characterization of the mucin or the mucus used in the studies was always crucial. What we found a little bit difficult is to define what is a muco-inert system and what would be an appropriate positive control for all the carriers at the same time, because some of the technologies would require a very stable labeling. In the case of the FRAP, it would also need to be photo-bleached, so it was not really trivial. And then finally, our main conclusion from this is that for stability and for colloidal evaluation of the carriers, we could adapt technologies for all the prototypes at the same time. But in the case of the mucoadhesion and mucodiffusion, there is no one single technique that could enable the comparison of all these prototypes at the same, in the same system. Yeah, I think my time is up. Just to say that the final conclusion from this work package has been the identification of 13 prototype families, and these are the ones that moved forward work package 2 and the subsequent work packages. Okay.